Hello, everyone. My name is Simon Verland, and I am a graduate student at the New York Botanical Garden and part of the team working on the living orchid collection of the Missouri Botanical Garden and its partners in Africa and Madagascar. Before we begin, I'd like to thank the World Orchid Conference organizers as well as the Taiwan Orchid Growers Association for inviting me to speak today. As you can see, this presentation is the result of the work of many people: botanists, engineers, technicians, students, etc. I'm the one presenting today, but I'm just a small gear in the machinery. With an estimated 27 to 28,000 species distributed around the globe, the word that could characterize best orchids is diversity. But how can we tackle this diversity, especially in the urgency caused by habitat and biodiversity loss? In Central Africa, our observations are that around 70% of orchids are epiphytic, and that at any given moment on the field, 90% of the orchids are not flowering, so cannot be identified all the way down to the species level. Historical dry collections are poorly preserved, and most often, critical characters for identification are not accessible anymore or destroyed because of the drying process. To make matters worse, because easily accessible forests have already been over-collected, and because of deforestation, the more or less intact forests are now days away from the nearest major city. Obviously, we can't visit on site regularly to gather flowering plants. So there is a need for an efficient and cost-effective procedure to sample the orchid flora of a given habitat. The solution we are implementing in Africa and Madagascar is to have a living collection hosted in a shadehouse. Think of the shadehouse as the tropical equivalent of a greenhouse. It's a very simple structure, wooden or metallic. It is very cost effective, each shadehouse costing only a few $1,000s to build, and they are very easy and cheap to maintain, especially if it's a wooden structure. Metallic structures, even though they are a little bit more expensive, tend to stay up longer with less maintenance. Also, because it's a simple and DIY structure, we can make all sizes down to very small shade houses, less than 100 square foot, like the one you see here in the middle in Antananarivo. A quick history of the collection with a few selected key dates. The cultivation of orchids in a shade house was first developed in Gabon by Jean-Philippe Biteau. He trained Tariq Stevar, who in turn implemented the technique in São Tomé in 1997 for his master's program. 3 years later, the São Tomé and Príncipe Orchid Guide was published. In 2001, a new collection was developed in Chimbele in Gabon, a biodiversity hotspot in Central Africa, complementing the existing collection of Mr. Biteau there. Still in Gabon, 2 other shade houses were also built in the Seabank Arboretum in Libreville in 2008 and 2009. In 2004, in Cameroon, the Yaoundé Shade House, our largest shade house in Central Africa, was created on the grounds of the University of Yaoundé 1 and since 2015 also hosts the first orchid seed bank in tropical Africa. In 2010, the Shade House network expanded to Madagascar with the start of our collaboration with the Mbatuvi mining concession, where 2 wooden shade houses were installed. In 2014 and 2015, 2 additional shade houses were built in Antananarivo and Umbalabe respectively. To date, the network currently counts 9 large shade houses in 5 countries with 2 seed banks, one in Cameroon and one in Madagascar, and around 25 people, yielding high-quality liquid specimens, pictures, and more recently, leaf tissue for phylogenetic analysis as well as root tissue for cytological analysis and the study of mycorrhizae. Here is our current network represented on a map. From our Nimba shade house in Guinea in West Africa, our shade houses in São Tomé, Gabon, and Cameroon in Central Africa, all the way to our shade houses in Madagascar. So how does the living collection work? First of all, we target threatened habitats in order to investigate the existing endangered diversity before it's lost. While on the field, we collect the most comprehensive sampling of orchids. These living plants are brought back to the shadehouse to be cultivated. Flower and leaf tissue are collected to make high-quality herbarium specimens, and the flowers are also cross-pollinated so that their seeds can be stored for long-term conservation in seed banks. The specimens and the information they provide are used in our conservation work, allowing us to identify the most threatened species. Distribution data feeds in our biogeographical analysis, helping us locate new high-value conservation zones, and of course, this trove of new specimens brings its share of taxonomic novelties. All this newly produced information feeds in conservation action plans advocating for habitat restoration, further inventories, and identifying new habitats to protect. And the cycle goes on. To date, more than 32,000 living, field-collected orchids have been grown in the living collection, representing more than 500 species. That's more than 280 species in Central Africa, around half of the orchid flora there, and 220 species in Madagascar, a bit shy of a quarter of the orchid flora of the island. Since 1997, more than 24,000 specimens have been collected, associated with silica gel preserved material and photographs. 41 taxonomic novelties have also been identified and described. More species potentially new to science have been identified and are awaiting publication. So what do we do with this volume of information and specimens? First of all, we are able to review the systematics of the orchids in these regions by working on their taxonomy, describing new species, for example, and by working on the phylogenetics, the relationship between species, with the help of molecular data. With plants easily accessible and with camera traps developed by our team, we can study the reproductive biology of these orchids and specifically identifying their pollinators, of which we know very little. Here is a Malagasy sphinx moth, and here is a still from a video captured of a moth visiting an African Sirtorchis. Finally, with the new distribution data we gather, we can also evaluate the level of threat the species is facing by assessing them with the IUCN Red List categories and criteria, identifying critical zones of high levels of endemism, or conducting broader biogeographical analysis to determine the geographic history of these orchids. By now, I've already mentioned a few of the partners that make this collection a reality. First and foremost are our scientific partners. Two examples here. The first in Cameroon with the partnership between the Missouri Botanical Garden, the French Institut de Recherche pour le Développement, and the University of Yaoundé. This Cameroonian shadehouse is not only the largest in continental Africa, but it's by far our biggest training hub with 3 PhD dissertations and 7 master's theses based on this collection. In Madagascar, the partnership between the Missouri Botanical Garden, the University of Vienna, and the Zoological and Botanical Park of Tsimbazaz in Antananarivo gives us not only the opportunity to host plants right where the research is being done, but also to have threatened plants at the ready for cross-pollination and safekeeping of the genetic material in our seed bank. The industrial partners are also critical for our collection in terms of the scope to which research can be conducted on these sites. In Guinea, West Africa, our partnership with the Société des Mines de Fer de Guinée, a mining project in the Mount Nimba range, has allowed us to gather around 1,000 plants from within and outside the concession and describe new species such as this lovely Polystachya orophylla. In Madagascar, the partnership between the Missouri Botanical Garden and the Mbatuvi Mining Company, the largest mining operation in the country, has made it possible to gather thousands of samples and make this small portion of the eastern escarpment forests the most documented in terms of orchids on the island. Finally, also in Madagascar, the Missouri Botanical Garden has partnered with the local community to manage one of the newly protected areas of the island, the Umbalabe Forest. A shadehouse was created and is managed by a team of trained villagers with the help of a Malagasy botanist from the Missouri Botanical Garden in charge of the oversight of the collection. Our sampling effort in this forest resulted in the recovery of several threatened orchids because of logging, slash and burn agriculture, but also the local and international horticultural trade, such as Sambidiella pardalina and Cryptopus paniculatus, both evaluated as endangered under the IUCN Red List categories and criteria, but also rare and not well-known species such as Bulbophyllum mirificum, described only 20 years ago. This partnership permits us to fully integrate in situ conservation with the protection of the forest as a whole, and ex situ conservation with the shade house hosting a comprehensive sampling of the orchids found there. A few words on our outreach and dissemination efforts. First, towards the scientific community, with numerous peer-reviewed papers, our red listing efforts, but also posters and presentations in international conferences such as this one. We also created a web-based platform called the Epiphytic Orchid Projects, using Tropicos, the online database of the Missouri Botanical Garden, and the Madagascar Catalog, the foremost repository of information on Malagasy flora. On this platform, relevant data such as current identification, locality, and ecological information of both cultivated plants and collected specimens is stored. The Epiphytic Orchid Projects is our attempt to deliver an authoritative synthesis of tropical orchids. Towards the general public, let me mention the Central African Orchid Database website, where people can find information on our work, but also a comprehensive picture collection of our cultivated orchids in Cameroon, along with taxonomic identification keys and relevant bibliography. Also important are our articles in the American Orchid Society's publication, Orchids. The shade houses themselves are a great tool to build and increase awareness on orchids and the threats they face. From high schoolers to politicians and decision makers, people can get up close and personal with the plants while visiting, allowing them to connect more easily. As I mentioned earlier, the living collection has proven its effectiveness in capacity building with several PhDs and master's thesis work undertaken under its roof. But I also like to stress that a conscious effort was and is done towards building local capacity. Local botanists and students are constantly improving their and our knowledge of African orchids. With its long-term involvement in the free African hotspots, that's Central Africa, Eastern Africa, and Madagascar, the Missouri Botanical Garden is key in training the next generation of African botanists. We are a diverse and young team that I'm proud to be a part of. I'll finish my presentation with a few key points that I hope I've made today. The Shadehouse Network and the Living Collection are a critical tool for the exploration and description of the orchid diversity, the ex situ conservation of the most threatened species, the outreach to local communities as well as the broader public, and finally, the training of the next generation of botanists working on orchid. I would like to thank you for your attention. I would like to thank again the World Orchid Conference organizers and the Taiwanese Orchid Growers Association for making it possible to speak today. I would like to thank our numerous partners for making this endeavor possible. And lastly, I would like to thank my team for helping me with this presentation. Thank you very much.